Welcome everyone to the afternoon session of our symposium. It's my great pleasure to introduce you Dr. Gabriel del Rio. Dr. Gabriel del Rio received his PhD in biotechnology from the Instituto de Biotecnología UNAM in 1997. Since 2004, serves as a full professor at the Department of Biochemistry and Structural Biology in the Instituto de Fisiología Celular UNAM. Dr. Del Rio is broadly interested in contributing to the development of new antibiotics in Mexico. His group studies the structure-function relationship of both the molecules with antibiotic activity and their targets. That is why his group studied the structure-function relationship of antibiotic peptides and proteins, in particular those peptides or proteins that are multifunctional. Dr. Del Rio uses computational approaches based on machine learning and other experimental models, including the yeast, Saccharomyces cerevisiae, and other cell types. Today, we are here to listen to his talk on the use of computer modeling in the identification of antimicrobial peptides with novel mechanisms of action and therapeutic applications. Thank you, Dr. Del Rio.
My name is Gabriel del Rio. I will talk to you about a story that I've been building in the past 20 or so years. This story is about how to develop antimicrobial peptides. And for that, I want just to briefly summarize what we know about antimicrobial peptides so far. These are mostly synthesized from the ribosome. There are some few which are synthesized by other means. And most of them, the reported ones, are chemically synthesized by humans. So we try to understand how they work, and so we chemically synthesize them. This is what you can see on this part of the image. Then the histogram shows you the distribution of these peptides. And in the bottom panel, you will see that most of the antimicrobial peptides identified so far come from animalia. And this is an important observation, considering that in animals, antimicrobial peptides may have more than just the antimicrobial activity. They will have other roles, such as immunomodulation activities. The antimicrobial peptides identified so far present all different kind of secondary structures, as it is shown on the right side panel. Most of this information, you can see it on the links that are beneath the image. One of the first things that we did to learn how to identify antimicrobial peptides was to understand how the mechanism of action of these peptides work, and the image on the left side will summarize the three modes of actions of these peptides on membranes. In all these cases, you will see a complementarity between the peptide and the membrane. Usually this complementation is derived from the charge, and many of the antimicrobial peptides are cationic, and some of the bacterial membranes tend to be electrophilic, and that induces the complementation. But also, these antimicrobial peptides were capable of traversing through the membrane, and that required the peptide to be amphipathic as well. So it should have a hydrophobic side and a hydrophilic one, which is cationic in this case. Considering these properties of the known antimicrobial peptides back in 2 decades ago, we learned how to predict these using physicochemical properties such as charge and hydrophobicity, and among other ones. With those criteria, we learned that a neuropeptide called substance P was also an antimicrobial peptide. And this is what I'm showing you on the right side of this panel. On the top figure, what I'm showing you is the activity against E. coli cells of this substance P at different concentrations. And on the bottom panel, what you will see is the non-toxic effect of substance P against a line cell that doesn't express the substance P receptor. Back in those years, there were more than 12,000 publications of substance P. None of them reported any antimicrobial activity for this peptide. And this is something that we learned. And eventually, the discovery led us to identify the first cell death mechanism mediated by autophagy. But that's a different story that I won't talk to you today. The story is just to explain to you how we learn that some antimicrobial, some physicochemical properties are sufficient to identify antimicrobial peptides. And that led us then to wonder what was the distribution of these physicochemical properties. And so in order for us to compute all these physicochemical properties, we implemented a device which is called an FPGA, which means Field Programmable Gate Array, which allow us to compute in, let's say, real time. We didn't need to have a supercomputer with a cluster of computers running for months. The 5 physicochemical properties that we use to describe antimicrobial peptides. So in the left side, what you will see is the general design of this FPGA, where we computed the physicochemical properties. And on the bottom part of this image, you will see how the values that we obtained for 2 of those values, how were distributed. You will see on the panel A, on the bottom image, that the isoelectric point is not a continuous, it doesn't have a continuous distribution. And on panel B, you will see that hydrophobic moment actually does have a continuous distribution. So these results led us to propose that in order for us to identify antimicrobial peptides, it will be more successful if we just copy the values of some of known antimicrobial peptides than to try to identify a particular range of values because of this discrete distribution that we observe on the isoelectric point. And considering that, then we put on a test this idea and define a concept that we call the physicochemically homologous peptides. So, and that idea is basically what I just explained to you before. We are trying to mimic the physicochemical properties of known antimicrobial peptides. And for that, we chose 2 of the most selective antimicrobial peptides reported back then, which were secropin and meganin. By selective, what I mean is that these are active against bacteria, but they display very low toxicity against mammalian cells. So using these criteria and the same physicochemical properties that we identify early on, identifying the substance P as an antimicrobial peptide, we designed a dozen of antimicrobial peptides, and all of them turn out to be antimicrobial with selectivity, which in some way supported the hypothesis of these designs. Among these peptides, one of them was a very interesting concept that we wanted to test that is related to what is referred in physics as the emergent properties. So an emergent property is that is not simply the sum of the features of the components of the system, The way in which I'd like to explain this is summarized in the image that is in the bottom left part of this slide, where you see a hand that is like a fist. This is a particular structure, and a molecule that will have this structure. Its function might be to knock on the door. Then you have another hand that is an extended hand that this is its structure, and its function will be to say hi, by waving the hand. Now, if we want to make a molecule that will have these two properties, that is to knock the door and wave and say hi to another person, what you will have to do is to induce intermediate conformations in this hand to pass from one conformation to the other one. And in doing so, it might be happening that if in the context where these transitions of conformations happen, a pencil appears or a pen happens to be in front of the hand, then the hand can grab the pen and start writing. Writing is not the sum of knocking the door and saying hi with the hand. It's an emergent property. So we hypothesized that by combining two activities in a single peptide molecule, we should have emergent properties. So how we did that? Well, the peptide the family of peptides that we designed is summarized in this right side of the panel. What you see on the red letters is the sequence of the alpha pheromone of Saccharomyces cerevisiae. These yeast cells are capable of mating by sex. And Saccharomyces is a heterothallic organism, meaning that it has two different cell types for mating, MAT-A and MAT-alpha. Each one of these cells secretes a pheromone. MAT-A secretes the pheromone A, and MAT-alpha secretes the pheromone alpha. And each one of these cells also produces on the surface of the cell a receptor for each one of these pheromones, and that induces the mating process in these yeast cells. So we have in red letters here on the right side of the image, the sequence of the alpha pheromone that is inducing the mating on the MAT-A cells. And this peptide is not an antimicrobial peptide, but we thought, since we have a ligand for a particular cell, we can make this ligand peptide into an antimicrobial one by adding amino acids on each one of the terminus of this pheromone sequence. So we added amino acids on both sides, and each time that we added an amino acid, we question, is this sequence an antimicrobial peptide? That antimicrobial peptide should match the secropin or meganin physicochemical properties. So the sequences that we identified to mimic any one of these two known antimicrobial peptides, all of them had 6 amino acids on the N-terminus of the alpha pheromone. And all of them mimic exactly the secropin antimicrobial peptide. So we identified 30 peptide sequences with those physicochemical properties, synthesized 4 of them, and were tested from a total of 344 million sequences that were generated by a computer. And in testing these sequences, we learned that indeed all of these sequences kill selectively the Saccharomyces cerevisiae MAT-A cells, but they didn't kill the MAT-alpha cell. Now, what was interesting for us is that the pheromone alone, or the peptide alone, or if these peptides were supposed to inactivate the mitochondria of the MAT-A cells, and this is because the the pheromone will be internalized by the MAT-A cells. And once that this antimicrobial peptide is inside the cell, may recognize the mitochondria as a bacterial membrane and will destroy this membrane, killing the cell. Well, the pheromone, or having a yeast cells without a functioning mitochondria, is not capable of killing yeast cells. However, these peptides that we refer to them as the Ishli peptides were able to kill the cells, and this activity was identified as an emerging property. To further characterize how this peptide was killing the alpha pheromone, the yeast cells, and these results were generated by Jonathan Rodriguez, which is in the pictures on the top side of this image, of this slide. Then Christian Diener, which might be familiar with some of you because he did a postdoctoral stay in Mehen some years ago. So he worked together with Jonathan to characterize how this was happening. And so the first thing that they studied, Jonathan did a microscopy analysis to show that the Ishli peptides were able to be internalized by several types of cells, including Saccharomyces cerevisiae, other fungi, as well as mammalian cells. Then Christian wondered how the peptide might be killing, and he consider two possibilities. The first one is that the peptide will be internalized directly by the cells, and inside the cells will then destroy the mitochondria. But then the alternative will be that the peptide will be recognized by the receptor, and once that is recognized by the receptor, it will separate from it, but being close now to the membrane could be internalized. So what Christian discovered is that the first model, the one that doesn't require the receptor, sorry, it does requires the receptor, but it's internalized through endocytosis. The second one doesn't require endocytosis. Behaves like the red line depicted in this right side image. But the actual data, which is seen in this image with the black circles, is best described by the second mechanism. That is a mechanism that doesn't require endocytosis. We did experimental testings of this idea, and we were able to show that there was no endocytosis mediating this mechanism of action. So this second model seems to be explaining how the Ishli peptides work, and these reported for the first time a novel mechanism of action for antimicrobial peptides. Mechanisms where two independent, apparently, mechanisms are playing a part. One that is receptor-mediating interaction, and a second one that is direct internalization by the membrane. So these results then led us to question how come that a peptide that is working through a novel mechanism may have an emergent property. What is the emergent property that allows the peptide to kill a cell? And by trying different experiments, among other ones, checking on null mutants on yeast cells, we learned that these peptides activated a mechanism that is referred as autophagy. Autophagy, as I explained to you, is something that we learn by discovering the antimicrobial activity on substance P. And so Ishli peptide, another antimicrobial peptide, also is capable of inducing autophagy. And in order to prove this idea, Erika, which you see on the top side of this image, of this slide, what she showed is that indeed the peptide, the Ishli peptide 1, which is one of the 4 that we tested originally, is capable of inducing autophagy in mammalian cells. As you see on the left side of the image, you see a huge staining, the nuclei staining of mammalian cells in blue, and on green dots, what you will see is the staining of the autophagosomes. In the presence of the Ishli peptide, you see an increased number of these green dots, not as much as a positive control of autophagy, which is 3 allos, but at low concentration, but a higher concentration, it reaches about the same autophagosome in number of autophagosome in these cells. Erika also showed that the isoforms of LC3 and a protein involving autophagy also changes. And she also showed that the autophagy is a continuous process by 2 different experiments. One is the degradation of P62, which is a protein that is part of the machinery of the autophagosome. And if the autophagosome is degraded by the back wall or the lysosome in the case of the mammalian cells, the P62 needs to be degraded. If the cells are only forming the autophagosomes, then P62, it won't be degraded. And the second one is the localization of the 88 protein, which is also shown to change color because it is going into degradation. So how is that the peptide is capable of inducing autophagy? Also through many different experiments, we end up discovering that the peptide is capable of sequestering ATP. What I'm showing you on the bottom part of this slide is on the left side, the in vitro quantification for the affinity of the peptide for ATP. The affinity is in the order of 400 micromolar, a concentration that is not reached outside of the cell, of the mammalian cell. So just for you to know, mammalian cells secrete ATP outside of the cell, and the concentration outside of the cell of ATP is very low. It's lower than 400 micromolar. But then inside the cell, the ATP concentration is in the millimolar concentration. So to prove that this affinity is effective in vivo, what Erika did, she ran 2 experiments, well, in collaboration with Lourdes, the group of Lourdes Macié at the Instituto de Fisiología Celular. So what they did with us is to check what was the effect on the ATP levels by putting the peptide either outside or inside the cells. And what we discover is that only when the peptide is inside at, let's say, high concentration, which is 50 micromolar, we were able to detect a reduction in the level of ATPs. In the others, we didn't see any effect. And so the peptide is only toxic at high concentration of ATP, of the peptide, of the Ishli peptide. So these results indicated that the peptide has an activity that is reflected in vivo. And this could explain how the peptide may be toxic to cells at high concentration, but at low concentration, which is at the concentration where the peptide induces autophagy, we suspect that, because we don't have a proof yet of that, that the peptide may be locally sequestering ATP that is the one responsible for inducing autophagy. So what I've been telling you so far is that we have an Ishli peptide capable of inducing autophagy on one side, but at the same time is able to kill microbial cells. So this will be, to the best of our knowledge, one of the first antimicrobial compounds that will be capable of regulating an activity that is important for the immune system in mammalian organisms. So this image is just to explain you what happened in the case of a bacteria that is been infecting humans for thousands of years, which is the Mycobacterium tuberculosis. When this bacteria is residing outside of the cells, the immune system will destroy these cells. So in order for these cells to escape the destruction from the immune systems, the cells are internalized inside macrophages. These are immune cells. And once that the cells are inside the macrophages induces autophagy to destroy these cells. However, the Mycobacterium is able to inhibit the degradation of the autophagosomes by the lysosomes. And then, thanks to that, it may survive inside these cells without being touched by the degradation machinery of the cell, and also escaping the immune cell. But if an infected cell like this one, you induce autophagy on that cell, you can get rid of the infection. So we thought, since this peptide is able to induce autophagy and also to kill bacteria, perhaps this might be a way to get rid of Mycobacterium tuberculosis that is resistant to either antibiotics or the immune system. So we thought or hypothesized that this peptide might be a solution for multi-drug resistant infections. To test this, Jacqueline, which you see on the right side of this slide, she performed several experiments. What you see, the first experiment that she did is the evaluation of the activity, of the capacity of these, peptides to kill the Mycobacterium tuberculosis in isolation, that is in culture. So she actually showed that the peptide is capable of killing either the drug-sensitive strain of Mycobacterium, as well as the multi-drug resistant strains of Mycobacterium. But then she also showed that the peptide may kill the bacteria that resides inside macrophages that are infected with this bacteria by activating autophagy. And what you see on the right side panel, which is perhaps the most interesting results that Jacqueline in the group of Rogelio Hernández Pando at the Instituto Nacional de la Nutrición here in Mexico City, is that we were also able to show that the peptide can alleviate the symptoms associated with tuberculosis in animals, in mice, infected with both strains, the drug-sensitive and the multi-drug resistant strains. What you see is on these right side panels is the lungs of mice that were infected but not treated with our peptide, and you see that the reduction of the areas where the lungs capture the oxygen that you will see like in blue, and in purple you see the staining of the cells, and then the lungs of these animals, you will see them once that they are treated with the peptide, how the lungs are capturing the oxygen or the air. So this looks like a normal, healthy lung, as well as the animals, right? So so far what I have told you now is that we learn how to design peptides by mimicking known antimicrobial peptides. But then at this time, we realized that the use of machine learning could help us to segment the areas in the physicochemical space where we could separate antimicrobial from non-antimicrobial peptides. And we could do that if we change the physicochemical descriptors at the same time that we use machine learning algorithms. So Jesus Beltran, which you see on the right side of this slide, implemented a method that allows us to select the physicochemical properties that describe antimicrobial peptide sequences to characterize the by either a random forest or an SVM or support vector machine antimicrobial peptides from non-antimicrobial peptides. And what he showed, and this is what you see on the right side images of this slide, is the efficiency for the classification of antimicrobials and non-antimicrobial peptides. So basically, he achieved close to, in the best case scenario, that is in the random forest, a scenario close to 94% of, or 95% in the case of the support vector machine for one particular data set. But in most of the cases, it was above 80%, which with these results, what he showed is that this is the best, this is the state-of-the-art classification method for identifying antimicrobial peptides. So having this capacity to predict antimicrobial peptides, now we can start doing many different things, things that I haven't mentioned so far, and I won't describe in detail, but I just want to give you this example for you to think about it. What we were able to show is that we can train with this approach, machine learning based approach, a machine that was able to learn from peptides to identify compounds, non-peptidic compounds that will have an antimicrobial activity. So this is the work of another student in my lab, Rodrigo Nava, where we were able to identify drugs that are sold for treating different human diseases that are not infectious diseases that will have as a secondary effect an antimicrobial activity. So, okay, so this is one of the many different things that we've been doing. Once that we know that we can predict or classify antimicrobial peptides with this efficiency. But there are many other things that we can do that I won't be talking today about it. So just to conclude my talk, I just mentioned to you that that the antimicrobial activity of peptides is perfectly associable with physicochemical features of peptides. Among these, we learn that a family of peptides that we refer as Ishli peptides will have a strong emerging property, that is, if we combine the pheromone binding activity with the antimicrobial activity, they will display an autofungic activity that the two independent activities won't display. And this antifungal activity seems to depend on the ability of these peptides to bind to ATP, which in turn induces autophagy or became a toxic substance for the cells. And then I finally show you briefly, unfortunately, but if I'm open to your questions, that the combination of an antimicrobial activity with a pro-autophagic activity is an effective way to treat resistant infections. Just to summarize, to finally thank all the people who have contributed to these results. The UNAM of the Universidad Nacional Autónoma de México, the group of Susana Castro, Antonio Peña, Roberto Coria, Salvador Uribe, Alfredo Torres, Miguel Costas, Lourdes Macié, Iván Ortega, and Gerardo Corso. In CICESE, which is in Ensenada, Carlos Brizuela and Jesus Armando Beltrán help us to design this protocol for identifying antimicrobial peptides. At the Instituto Nacional de la Nutrición, the group of Rogelio Hernández, working with Jacqueline, Octavio Ramos, and Dulce Mata, help us to validate the usefulness of antimicrobial peptides with autophagic activity to treat resistant infectious diseases. And in the group at the Humboldt University in Berlin, the group of Eda Klipp and Andreas Hermann help us to characterize the mechanism of action of internalization and action of the Ishli peptides. The agencies that have supported this work are the UNAM, Instituto de Fisiología Celular, CONACYT, and the Alexander von Humboldt Foundation. So I appreciate your attention, and I am open to your questions.
welcome Dr. Alejandra Cervera Taboada. Dr. Alejandra Cervera Taboada is a researcher and coordinator of the Population Genomics Division at ITMEGEN. She got their master's degree and PhD in bioinformatics from the University of Helsinki, Finland. Her research interests are genomic profiling and searching for biomarkers for cancer. Additionally, she has developed several bioinformatic tools to identify key players in cancer regulation. Today, we are here to listen to her talk on molecular classification in pediatric PO, the genomic impact on clinical practice. Thank you, Dr. Cervera, for being here.
Hi, good afternoon, everybody. First, I want to thank the organizing committee for inviting me to talk to you about the project I've been working on with the Genomics Diagnostic Lab here in ITMEGEN, which is molecular classification in pediatric B-cell acute lymphoblastic leukemia. First, as an introduction, acute lymphoblastic leukemia is the most common type of childhood cancer. B-cell acute lymphoblastic leukemia is the leading cause of cancer-related deaths in children and young adults. And this is a cancer that's characterized by recurrent genetic abnormalities. These genetic abnormalities can be used both for prognosis and for guiding treatment. While 5-year survival rates have improved drastically in many countries, unfortunately in Mexico, it's still around 62%. And there are many causes for this. There are two main groups of causes, perhaps. The genetic ones is that in Mexico and also in Hispanics in the US, it's been observed that there's more patients with poor prognosis. This is due to higher frequency of poor prognosis genetic alterations or because they are older at the age of diagnosis. Also, likewise, we observe less patients that have these good prognosis genetic alterations. And the environmental causes that can be causing this disparity in survival, of course, may be due to lack or delay in diagnosis, misdiagnosis, obstacles to accessing care, abandonment of treatment, death from toxicity, and relapse. So this is a project that's been sponsored by the Health Ministry in Mexico, and the idea is to try to help improve diagnosis and also to assign patients to the right treatments so we can lower the death from toxicity in these patients. The overview of how this disease is usually diagnosed and how the molecular classification is achieved is that first there is a bone marrow biopsy, and then the pathologist determines that this is in fact B-cell acute lymphoblastic leukemia. For deciding if which molecular subtype we have, then usually karyotyping, FISH, or RT-PCR are used, and this helps us see if there is gains or losses of chromosomes. If we identify this BCR-ABL fusion, which is known as a Philadelphia chromosome, and then we look for this other group of fusions. And finally, we looked also for amplifications in chromosome 21 and rearrangements of this particular gene, formerly called MLL, now KM2TA. And the patients that don't get classified with these assays, then usually are further explored again with RT-PCR or amplicons or next generation sequencing. And mostly what we look for these patients are for alterations that are associated with what is called the Philadelphia-like phenotype. So patients that have a similar phenotype to the BCR-ABL1 fusion, but that they in fact don't have this fusion. So there are specific fusions associated, specific mutations, as well as overexpression of CRLF2. Our diagnosis strategy, or what we're doing in this project, is try to simplify all those assays into using one RNA-seq gene panel. So the idea of not using whole transcriptome is to lower costs, and using RNA-seq perhaps allows us to classify the most patients with one test. So, for our project, what we have right now is 113 samples, both from historic and new patients. The gene panel consists of around 1,400 genes. And what we have been doing is fusion and variant calling, plus gene expression profiling of certain genes, because we don't have whole transcriptome for these patients. Here I want to share with you the mutation and landscape of our patients. These are only looking at pathogenic and likely pathogenic variants, so it's not all the mutations that we have been finding, but the ones that are known to be deleterious. And here, consistent with what's been reported in literature, we see a lot of mutations in the Rnas gene family, and we also see a lot of pathogenic mutations in a lot of cancer-related genes. Specifically relevant to clinic is that the CRLF2 mutations that we have been observing are known to sensitize patients to a specific JAK inhibitor. So this mutation analysis, it's very helpful for guiding treatment. As a second step of our pipeline, what we're doing is identifying fusion genes. And here we can see that the most prevalent one is the TCF3PVX1, then followed by FV6RONX1. This is actually the opposite of what happens in reported literature and in other cohorts, especially in Caucasians or white people, sorry. What we observe in those cohorts is that this is the most prevalent one and this is the second one. Then we here have the group of fusions that have been associated with what is called the Philadelphia-like phenotype. Here we have the Philadelphia fusion. Here we have a big group of patients that have fusions that are not categorized yet. So this is a group that doesn't exist yet. What we see here is some players that are already emerging in the literature, like CREP-P and PAX5 as important drivers, but they are not yet an official category yet. And also we have MF2D and this KM2A fusions, as well as other fusions involving FP6. So with this analysis, we managed to classify about 40 patients, but we still have around 73 patients that are not classified yet in any of these molecular subtypes. So what we have been doing next is to try to identify this CRLF2 overexpression. This in the clinic is usually done with RT-PCR, and for people that have access to whole transcriptome RNA sequencing, they can do TSNE, or some people just take the top one expression, samples of the expression of CRLF2 in order to categorize patients into this molecular subtype. What we have been doing in this project is using the K-means algorithm to try to find different clusters of expression. I think this has worked very well for us because we can see that we have a gap here in between these three groups of expression and the highest expression group. So next, what we have been doing is to try to identify what molecular mechanisms are driving this overexpression here. And here I'm going to show you the same plot, but now we have the samples with their own categories here. And what we can observe right now is that all of these events are related to two, except for these three over here. And the one that is the most predominant one is internal duplications, or CRLF2. Next, what we observe is that this fusion gene is here in these two groups, but the difference between these groups is that this one, apart from the fusion, it has also a mutation. So what we think is that actually it's the mutation that is driving the overexpression here, not as much as the fusion. This is also consistent with literature, but then the obvious question here would be, if it's the mutation, why do we have a mutation in this cluster that is with lower expression? And the explanation here for us is that the mutation is actually in low frequency in this patient. So it's not strong enough to drive the expression all the way to the C4 cluster. And finally, this other one, fusion that it does not include the CRLF2 gene, but it's a known fusion and it has already been associated with this particular phenotype. So I think that K-means algorithm helped us really separate the groups here, and we have then managed to classify more patients. And here I'm showing you the classification of the patients as well as the prognosis for these different classifications. So like I mentioned before, 8p6 RONX1 is the most recurrent fusion in other cohorts. It has a very good prognosis. And unfortunately for our samples or our patients, we have a much lower number of these patients. And we have more of this DCF3PVX1, which has a worse prognosis than this one. And also we observe that we have a lot more of patients that have Philadelphia-like phenotype and that unfortunately have the worst prognosis. So in our cohort, it seems to be consistent with what has been reported that Hispanics have higher incidence of leukemia and also have worse survival rates due to just a genetic factor independent of the other aspects. So after the serial 2 analysis, we had more patients classified, but we still have a huge number of patients that are unclassified. So what we have been working on is trying to figure out what's happening with these patients and if there's any way that we can identify what classification they could have or what better treatment it's more suited for them. So in that sense, we found that some patients had a gain in MED12, and when we use again K-means algorithm with the different classifications, we realized that most of the patients that have this gain actually belong to the unclassified group, except for this Philadelphia-like patient here. It seems that a lot of these patients don't have a category yet. So we thought this was very interesting and worth looking deeply into it. So what we did next was differential expression analysis between the lower expression group and the highest expression group. And I think that the clusters look here really defined, and this is quite surprising because this is, remember, a panel of genes, so we're not looking at the whole transcriptome. And what we observe here is that this group and this group that have the high expression of MED12 actually have high expression of all these genes. And what we notice from the allocation of these genes is that it seems that several of them are in the same region of chromosome X. So we're thinking now that probably it's not just MED12, but it's again in this whole section that is driving this phenotype. This still needs to be, of course, validated through DNA, but so far it seems to be the case. And then, of course, we would like to know if what we observe here is something that it's an artifact from our cohort or this is happening elsewhere. So for that, we look at the target cohort, which is a public data set of B-cell acute lymphoblastic leukemia patients. And surprisingly, we have a very similar pattern in which the majority of the samples that have the highest levels of expression for MED12 are actually unclassified. With the advantage of this data set, we can do now differential expression on the whole transcriptome, and here we have MED12, and here we have again SMIM3, which was a neighboring gene. We don't observe some of the other genes from the same region of chromosome X here, but they are in the list of differentially expressed genes. This is just a subset of those genes. And also, I forgot to mention earlier that MED12 and SMIM3 are genes that are quite far away from CRLF2. So if there is an amplification, it's an amplification in a different region. So it's not the same patients and not the same event. So we're thinking this is a little bit different. Now that we have the whole transcriptome, we were able to do some pathway enrichment analysis. And one of the pathways that I think it was the most interesting to look at was a positive regulation of apoptosis. So, of course, this is a little bit hard to interpret, because we see that we have upper genes in both groups of patients. TP53 is actually on this upper part. And to get a better understanding of if actually MEF12 overexpression or this phenotype that we are observing has an impact on the clinic or on patient prognosis, what we did next was some relapse-free survival analysis, and this doesn't look as pretty as I would like to. We have a big overlap of the confidence intervals. We think that maybe it is true that maybe patients with overexpression of MEF12 will have a better prognosis, but it is hard to see here because the target cohort, at least phase 2, which is the one I have been using for this analysis, is a high-risk cohort. So most patients here were expected to relapse from what was observed at diagnosis. So we think that maybe we had more patients and less biased cohort, maybe the separation here will be clear. This is, of course, work in progress, and we don't know as of yet if this will hold or if this would be actually relevant for the clinic. But this is something that we are currently working on. So apart from that, we want to increase our sample size of Mexican patients and then try to see if we can correlate these results with clinical data from these same patients. So currently, we don't have more patients. We don't have the whole clinical data because some of these patients have been just recently diagnosed, but we're also getting more samples from the historic data set. So maybe we can better correlate with the clinical data. And of course, also we're very interested in looking into the pathways and mechanisms behind the MEF12 expression groups to see if this actually has an impact on how to treat patients or how to better classify these patients. And for that, I need also to identify different cohorts that could help me with this. So lastly, I want to thank, of course, my collaborator, Carmen Alaez-Berson, which she has been very kind of trusting me with her data and inviting me to collaborate with her in this project. I want to thank everyone here for listening to my talk, and again to the organizing committee for allowing me to present my research. Thank you very much.
Thank you, Dr. Cervera, for your talk. We have time for questions. It is my great pleasure to welcome Dr. Raquel Cuevas as our last speaker of the event. Dr. Cuevas is a full-time research professor at the School of Medicine of Tecnológico de Monterrey and as a part of the Bioengineering and Regenerative Medicine group. In 2014, she obtained her PhD degree in biotechnology from Tecnológico de Monterrey in Mexico, working in the cell therapy research group. Her research involves characterizing the transcriptomic and epigenomic changes occurring during the cell differentiation in brain development and in neurodegenerative diseases. She has experience analyzing next-generation sequencing data, including single cell. Today, we are here to listen to her talk on the systemic analysis of transcriptional regulations in diverse cell types of the central nervous systems. Welcome, Dr. Raquel Cuevas.
Good day, everyone. My name is Raquel Cuevas. First of all, I would like to thank the organization committee of this symposium for their invitation. It's a pleasure to be here. I am a research professor at Tecnológico de Monterrey at School of Medicine. My research is focused mainly on regulation of transcription through diverse mechanisms. One of them is epigenetic mechanism. The other one is the long non-coding RNAs. So I will talk about today some of the projects that I am working with, that part of the research has already been published and other part is still in progress. So I want to start with this figure that I got from the National Human Genome Research Institute. It's a very interesting figure, but it's very depicting about what is transcriptional regulation. So in order for cells to modify the expression of their genes, there are diverse layers of regulatory mechanisms that need to take place. So here we see, for example, there's the architecture of the chromatin. The chromatin can be very compact or very relaxed, allowing transcription. Then the DNA is wrapped around histone proteins, and then these histone proteins have tails that can have diverse modifications, for example, acetylation, methylation, trimethylation. I mean, it's a very, there's even a histone modification code to see what are the possible combinations. There's the DNA can be methylated, and it can be regions of the DNA will be silenced with this mechanism. And then once we have open chromatin regions, then these regions can also be modified by regulatory DNA. I am very interested in regulatory regions that are called enhancers. And then these enhancers act as scaffolds, and they trap transcription factors, and then they approximate these transcription factors to promoter regions of genes that can be located far away from themselves. So this mechanism is also very important. So all of these regulatory mechanisms act in combination to modify the transcription that is going on inside the cell. And this can, afterwards, it will modify the cell in the cell we differentiate, will have some plasticity. For example, if we're talking about neuronal cells, the neurons will remodel themselves. But also there can be some detrimental regulatory mechanisms, for example, in neurodegenerative diseases, where the cells will start behaving differently as they should. So the first project I want to talk about is collaboration with the Instituto de Fisiología Celular of UNAM and the UAM, and we are studying differentiation of pluripotent stem cells into dopaminergic neurons. We know that these neurons are the neurons that are mainly affected in Parkinson's disease. So what we asked ourselves at the beginning of this project is, how are the cells differentiating? How can we can have an embryonic pluripotent stem cell, and then with the adequate induction media, this cell will become a dopaminergic neuron. So in the Instituto de Fisiología, they already mounted the induction protocol that it's already validated in vitro and in vivo with some monkey model of Parkinson's disease. But the differentiation is not really described yet. So what we did was we performed RNA-seq to obtain all the transcriptional modifications that are going on. But this RNA-seq is like the highest level. We want to know who is regulating who. So we also perform ATAC-seq. ATAC-seq is a technique that it allows you to see what regions of the DNA are open or accessible and which ones are closed or inaccessible for transcription. And then after combining these two methods with bioinformatic analysis, we obtain transcriptional profiles. We perform motif analysis in the open chromatin regions, in the promoters of differentially expressed genes. We focus on the long non-coding RNAs because, as I mentioned, these non-coding regions of the DNA are transcribed, but they never become protein. So these are their main functions are regulatory. They regulate transcription. They can bind proteins. They can bind other mRNAs. They can bind DNA. They can. They have a lot of functions. And then we also want to look at enhancers. Enhancers are regulatory regions of the DNA that, as I mentioned, they can act as scaffold, and they can bind transcription factors and approximate them to regions of the DNA that are far away. So we saw that in effect, the differentiation of these cells is if we group by hierarchical clustering, we can group the gene expression, and there are groups of genes that have similar patterns. So this means that there are mechanisms that are taking place in the differentiation process, and that they are either becoming, for the red color means that they are highly expressed. These genes are highly expressed in the day 0. That means when the cells are still pluripotent. And when they differentiate, these genes become downregulated. So the gene set enrichment of these genes is pointing to the pluripotency. So mainly these genes are allowing the cell to have this pluripotential identity. And then as they become differentiated, these genes, like for example, SOX2, OCT2, NANOC, they become lowly expressed, or they start becoming inhibited. Okay, so and we have the opposite trend. We have genes that are downregulated at the beginning, and then by day 28 of induction, they are highly regulated. And these genes are related to neurons and dopaminergic synthesis, the dopamine synthesis. And within these cells, there are a big number of genes that are differentially expressed, but many of those are link RNAs, and the link RNAs are very, very important in regulation of protein coding genes. So the top differentially expressed genes from day 0 to day 28 are link RNAs. So that made us question ourselves, what are these link RNAs making? What is their function in this regulation? So the link RNAs are, they have been started to be studied maybe 10 or 15 years ago. They're rather young, and they are, many of them, the majority of them are not described. Their functions are still unknown, and many of them, the annotation is not very complete. So we also have the link RNAs. They are categorized according to their position with respect to protein coding genes, because many of them are antisense, and by being antisense, they can block the polymerase and they can block the transcription, so they can inhibit protein coding genes. but they can also enhance the transcription of protein coding genes. So as I mentioned, their functions are very diverse. So we saw that the majority of genes that are differentially expressed of link RNAs, differentially expressed, are long intergenic RNAs. These intergenic RNAs, they are in an intergenic position and they are far away, more than 5 kb, any protein gene. So the only way that you have to assess their function is through making gene correlation of gene expression with the closest protein coding genes, which can be megabases far away. But there are some link RNAs that have been described in other tissues. For example, SOX1 OT. SOX1 OT is a link RNA. The name means overlapping transcript. And there has been a report of the regulation of SOX1 OT over one. These are already known, and that's why their names are already modified. The name of a link RNA should be made depending on the gene that it regulates, and then the class of the link RNA. This is an overlapping strand. It's in overlapping, and it's in the sense strand. Okay, and here we have, for example, antisense. FOXD3 is antisense. So we found many examples of link RNAs that both the protein coding gene and the link RNA were highly correlated, and both of them were differentially expressed. So this means that link RNAs are having a function in the differentiation to dopaminergic neurons. Perhaps this function is even more important than transcription factors that are already known. Okay, and then our next question was, which are the regions that have an open chromatin and which are the ones in closed chromatin? So we analyzed the data from the ATAC-seq, and we observed that at day 0 in pluripotency conditions, the accessibility to the chromatin was the highest. Why is that? So when we have accessible chromatin, this means that the DNA is prone to be bound by transcription factors. So transcription factors can act and they can initiate transcription. So the genes, when a cell is pluripotent, it has many possible roads. It can become a neuron, it can become an astrocyte, a microglia, endothelial cell. There are many possibilities. That's why the chromatin is open. And as the cell starts differentiating, it starts narrowing down the possible paths that it can turn into. And that's why we see closing of these chromatin regions. The majority of the accessible peaks were intergenic regions and intronic regions. And this validates the fact that accessible regions are enriched in enhancers. Enhancers are these regulatory DNA regions that can trap transcription factors and approximate them to promoters of genes. So we performed a gene set enrichment analysis on the accessible chromatin regions that we found at day 0 and at day 28. And interestingly, they match. The accessible regions in day 0, they are related to pluripotency, differentiation, stem cell division, whereas at day 28, they are related to neurogenesis, axons, synapses. Okay, so this all makes sense. So, and then when we combine the transcription and the accessibility information, we see that there's a correlation, a modest correlation between accessibility and transcription. But basically, all the regions that are accessible are not necessarily transcribed. Okay, that's important. There are other mechanisms that are also acting on transcription, but if you have accessible regions, they are more likely to be transcribed. So here we see some examples. PRDM14 is a gene, a protein. It's actually a transcription factor that is important for pluripotency. And we see that at day 0, it has accessible regions. Here we have the exons. There are accessible regions in the promoter And there are also some intergenic and intronic accessible regions. And these regions become unaccessible at day 28. That also makes sense because these genes will stop being transcribed. And the transcription of this gene is high in day 0, but it's mainly silenced at day 28. So this is a perfect example of how we can match chromatin accessible regions with transcription. We downloaded, so the interesting part about doing this bioinformatic genome-wide analysis is that there are many public databases, for example, ENCODE, PsycENCODE. There are many databases that already have performed these high throughput sequencing assays. You can download them, the ones that are more similar to your example, and try to compare. So here we downloaded the H9 and also a pluripotent cell, and we compare with DNAs. DNAs will also give us information about chromatin accessibility, and then they match. The peaks that we obtain for attack at day 0, they match with the H9 and H1. H1 is also another pluripotent cell. And then we have also these marks, the trimethylation and the 27 acetylation. So these ones also, delineate enhancers. So we see here that this is most likely an enhancer region. Okay, and then we have also the promoter region will also have those 2 marks. Okay, and we are showing those marks for neural precursor cells, which are similar to our day 28, and the substantia nigra, which are mature dopaminergic neurons. And then when we compare another loci, this is chorin. Chorin is a protein that is highly specific for dopaminergic neurons. So we see that there's chromatin accessibility in the promoter region at all the 3 time points, but there are some intergenic regions that they start gaining accessibility at day 28, and they were not accessible at day 0. So these become, these are potential enhancers. They are called putative enhancers. We are right now performing histone marks for these day 28 specific examples, so we can know exactly which ones are active enhancers. Okay, so our working model right now is we are integrating these data sets, RNA-seq attack, and we are adding chip sequencing of 2 histone marks to know exactly which ones are the enhancers that are active. Then we are working also on mutating these regions, these enhancer regions, with a CRISPR-Cas9 model and in utero electroporation. And we expect to see either differentiation not occurring, or we expect that these enhancers regions are participating in the differentiation. So we want to see what is their effect. We are also doing the same for link RNAs. The link RNAs, if we disrupt their function, then there will be an effect in the phenotype. Okay, so then I want to talk about another project that I worked on before, which is the identification of long non-coding RNAs or link RNAs in brain cells. So this was performed when I was a postdoc at UT Health in the Institute of Molecular Medicine. We isolate brain cells from a mouse model, and through immunopanning, we were able to obtain all the different cells of the brain. These were mouse brains. We have astrocytes, neuronals, oligodendrocytes, microglia. The oligodendrocytes, there are 3 stages of oligodendrocytes that coexist in the brain. These are the newly formed oligodendrocytes, the oligodendrocyte precursor cells, and the mature oligodendrocytes. So that will be important. And then the link RNAs are known to be highly enriched in the brain. So by this heat map, we are showing that the gene expression of the top differentially expressed link RNAs in the brain all of these are brain cells. These are our brain cells. And if we compare to other tissues, you see there's no expression of these link RNAs. So that's why if you're studying the brain, you need to study link RNAs because they have a very important participation in brain. And also link RNAs are known for being highly cell type specific. So here we see a perfect example of how these link RNAs are only expressed in OPC cells, in endothelial cells, in astrocytes, in neurons. Okay, so this is also very important. There are also some tissue-specific cells. That's why link RNAs exert a regulation specific to tissues or cell types. So the difficult part or the challenging part about studying link RNAs is that the majority of them are unknown. Their functions are not known. They have not been studied before. So the only bioinformatic method that we have, or one of the most common ones, is guild by association. Through guild by association, we are able to predict the function of link RNAs. So in this project, we wanted to see what were the function of these highly expressed, differentially expressed link RNAs. And what you do is that you correlate the expression of a link RNA in diverse samples. In this case, we had the brain samples and the tissues, samples from other tissues. And you need to make a correlation of the expression of that link RNA to all the protein coding genes. And then you get the ones that are highly correlated, and then you assign a function to a link RNA through the function that are of the highly correlated protein coding genes. So that's why that's the name of this analysis, is guild by association. You become a link RNA gets a function due to its association with protein coding genes. So if there's a high correlation with protein coding genes that are involved in neuron differentiation, then by association, the link RNA that has this high correlation to protein coding genes will also be involved in neuron differentiation. So by making that analysis, we were able to see that the highly expressed link RNAs in these brain cells were related to brain functions like gliogenesis, neurogenesis, neuron migration, synapses, axon, and so on. Okay, and then we also have, okay, then when we were interested in looking at the differentiation from neuron stem cells to mature oligodendrocytes. Why? Because oligodendrocytes participate in myelination, and myelination is a process that is dysregulated, that is, the synapses become unmyelinated or lose their myelination in a neurodegenerative disease that is called multiple sclerosis. So they have a very high relevance for this disease. So if you look at, we had these brain cells and we had like a time differentiation because we have the neural stem cells, and then we know the neural stem cells will become or will generate oligodendrocyte precursor cells, and then they become newly formed oligodendrocytes and then mature oligodendrocytes. So by looking at these 4 samples, we can make like a continuum and make an analogy with oligodendrogenesis. Here we obtain the link RNAs that are more specific or differentially expressed in this time span. And for example, this one, we named it Lync OPC because it's very interesting. You can only see it in OPC cells. It becomes downregulated in newly formed oligodendrocytes and almost absent in mature oligodendrocytes. So this means that this link RNA could have a function in oligodendrogenesis. And then we also have these other 2 candidates. If you see this, we generated link RNA de novo assembly. So these link RNAs are unknown, but we see there's a signal in our RNA sequencing samples, and so there's something in this locus, and that's why we named it like that. Okay, and by looking specifically at the Lync OPC candidate, we see that in the promoter region, there we perform ChIP-seq for a transcription factor that is highly relevant in oligodendrogenesis. This is OLIG2. So we know that OLIG2 participates in the differentiation of neural stem cells to mature oligodendrocytes. So we perform ChIP-seq to see where we had binding sites for these transcription factors, and we have a binding site for this transcription factor in the promoter region of this link RNA. So that validates our hypothesis. And then we also made a motif analysis, and of course there was a transcription factor binding site motifs in this region. There's also an ENCODE has a set of promoter regions, and there's a promoter region defined by ENCODE. Okay, and then we went to the functional assays, and we saw that there's by a luciferase assay, a luciferase reporter assay, we saw that this link RNA, if you knock down the link RNA, there is no differentiation to oligodendrocytes, or it's highly decreased. So look at, I'm showing here the expression of Lync OPC. As we made a differentiation in vitro now, not by the cells that we isolated from the brain, but in vitro we induced to oligodendrocytes, and as the induction is occurring, there's a high increase of Lync OPC, and then when you knock down the Lync OPC, by knocking it down, you see that these are markers of oligodendrocyte cells. They become highly decreased. And then by immunostaining, we see that there's no differentiation, or is highly decreased. That O2 is also a marker of oligodendrocytes. So you see in these images, there's the differentiation. By knocking down Lync OPC, the differentiation stops. So that validated our hypothesis, and that's why Lync OPC has a high importance in the differentiation to oligodendrocytes. Next, sorry, I'm going very fast, but we have very few time. The last research that I want to talk about is also a collaboration with the UT Health. And in this case, we were working with a model of spinal cord injury in rats. And we were interested in what happens with astrocytes. So when there's a spinal cord injury, astrocytes change their phenotype, and they become what is called reactive astrocytes. So they start growing, and they start forming, it's a astroglial scar. They start forming a scar in the injury, and that scar becomes detrimental because it inhibits the neurons, their synapses, they start regrowing. But when you get this scar, there's like a physical barrier that cannot rejoin the nerves. So this is called astrogliosis, and it's a detrimental. So we want to see what is happening to these astrocytes. Why are they becoming reactive after injury, and what is driving that change in phenotype? So we obtain in bulk and in purified astrocytes, we obtain RNA sequencing, and we look closely into the link RNAs, which were the link RNAs that were becoming differentially expressed in both the bulk tissue and the reactive purified astrocytes. And then we validated some candidates doing a knockdown, and we found that there's this CEB2OS is a link RNA that is acting on CEB2. It's already this function has already been described in another, but in astrocytes, it has when you knock down this link RNA, the astrogliosis decreases. So this is highly relevant because it could help to decrease these reactive astrocytes. So I will briefly discuss. We also found patterns of gene expression that were occurring after the spinal cord injury up to 3 months. So this is a acute and chronic spinal cord injury. And we see that they are related to neurons, others are related to immune system. So we can see all the functions that are occurring after this injury. And there are 778 link RNAs that are differentially expressed at all the time points. So by looking at those link RNAs and looking at which ones have homology with human, because remember this is a mouse model, a rat model, sorry, and some of these link RNAs are conserved. And we obtained several candidates, and the candidate that we thought was more promising was this one. You see, we have CEB2OS. This is the link RNA, is antisense. So it's being transcribed in the opposite region to CEB2. This is a protein coding gene. And it's very close. Actually, it's overlapping some of the one isoform of CEB. There are 2 isoforms. It's overlapping the first exon of one isoform, and it's transcribed in the opposite direction. When we look at the expression, RNA, we see that the CEB, both CEB2 and CEB2OS, they are changing, okay? You see, for example, CEB2 is increasing and CEB2OS is also increasing. So what we did was, okay, we already have these candidates, CEB2OS and CEB2. So we want to see who could be regulating CEB2OS. So what we did was to perform motif analysis in the promoter regions of link RNAs, of all the link RNAs that were differentially expressed. And we see that there's a high enrichment of STAT3. STAT3 has already been reported to have a function in spinal cord injury. So, and it's differentially expressed, it's indicated in red. And so this was our main candidate. So STAT3 could be regulating CEB2OS, and that could regulate CEB2 and have an effect in astrogliosis. So when we look at the combined expression of these 3 genes, we see that they are all increasing as the spinal cord after the time of spinal cord injury. So this makes even more higher, these candidates are even better, or it gives some confidence on our hypothesis. So this is our working model. STAT3 could be regulating CEB2OS and CEB2. There's also binding of CEB2. We validated this with ChIP6 of STAT3. And CEB2OS could itself be regulating CEB2. And this acts on astrocyte proliferation and scar development, okay? We validated this through a knockdown. As I mentioned, we knocked down CEB2OS, and we observed that after the knockdown in vivo, there was a decrease in the spinal cord, in the fibrosis, in the astroglial scar, okay? And there was a lower expression of CEB2, okay? And also a lower expression of phosphorylated STAT3. GFAP is a marker for reactive astrocytes. Okay, so that gives us some candidates that for having a therapeutic target that could be validated in the clinic. So what are the take-home messages that I would like you to take with you? Right now, it's not only a matter of looking only at RNA expression, okay? Right now there are, with all these high throughput sequencing technologies, there are many other levels of regulation that you should integrate into your analysis. it's not enough right now by saying, yes, these genes are differentially expressed, these link RNAs are differentially expressed. Okay, but why? Who is driving those expressions? And you can do that by integrating epigenomic data sets. You can generate them in the lab, or you can download them from databases. As I mentioned, there are many public databases that have made all of these high throughput sequencing assays. And this is especially important in the nervous system because the nerve cells are highly, they have this high plasticity, they are always changing. So it's very important to see who are the drivers of those modifications. How is this chromatin being remodeled? What are the histone modifications that are taking place? What are the regions of the DNA that are becoming methylated? And this is also important in neurodegenerative diseases because it has been demonstrated that hypomethylation or hypermethylation are maybe driving these many of these diseases. And also non-coding RNAs, and not only long non-coding RNAs. I only talk about those because those are my specialty, but there are also Rnas that are very important, and there are a lot of categories of non-coding RNAs that have regulatory functions. So you should also look into them. Enhancer regions are also becoming very important because they are acting upon regulation. They are directly regulating the transcription of other genes. There are some ACEs like Hi-C that can tell you what regions of the DNA are binding, what regions, which ones are having interactions. So all of these data sets are high throughput and they need bioinformatic programs and bioinformatic to do the integration. So that's very important. It's really needed. Bioinformaticians are needed to analyze all these high throughput data. And then also one important thing that I want to mention is that these regulatory mechanisms are highly cell type specific. And they have to be because that's why we have the same genome, but with different function throughout all our body. And that's basically because of these regulatory mechanisms. And even more, right now, doing bulk assays is not the best right now. Right now, you should go into single cell. So that's why we are we are also right now working on single cell of these neurodegenerative diseases. So you should really look into single cell right now because there's many things that are missed when you do bulk assays. Okay, and then this high throughput sequencing, as I mentioned, is becoming increasingly accessible. So you can either get them from public databases or you can sequence. It's becoming cheaper and cheaper. So right now, the challenging, I think the most challenging thing is integrating all of these data sets. Okay, and with that, I want to thank you, and I will be open, and it will be my pleasure to answer any questions that you may have.
Thank you, Dr. Cuevas, for the interesting talk. Now we move to the questions.
Hi, everyone. Before closing this meeting, I would like to recap and make some reflections around what happened in these 3 days during the symposium. In this virtual meeting, we have seen theoretical approaches to tackle some fundamental questions in systems biology and their application to understand wellness and complex human diseases. Here, we have seen the use of classical approaches, including constraint-based modeling, ODE, stochastic theory, network theory, and Boolean modeling of transcriptional regulatory network. All these frameworks I consider paradigms in systems biology now. With this formalism, in combination with machine learning techniques, we are getting more ambitious objective to potentially develop the precision medicine. These approaches are needed and valuable to discover the principle of life, paraphrasing Franz Fayakov, the logic of life. In terms of application, high-throughput technologies are impacting health research. Meanwhile, single-cell RNA-seq can be used to study Alzheimer's. Multiomics data approaches are contributed to move toward a holistic description of human diseases. As well, we have seen how genome population scheme and systems immunology frameworks are impacting melanoma studies. Also, genetic variables are contributed to the molecular classification of leukemia in populations. In the same direction, the microbiome, currently a possible new disruptive technology, has been applied to design personalized diets for improving and preventing some complex diseases, such as cancer, diabetes, or autism. Finally, but not least important, we have witnessed how physiological networks are contributing to the study of complex diseases and how the use of computational modeling is essential for identifying antimicrobial peptides with therapeutical implication. The spirit of the meeting is focused in one aim: to create a space to share knowledge between different groups of interested in human health and human complex diseases, and stimulate the discussion and collaboration between groups. This meeting is an effort to get these aims. Finally, we want to say thank you to all the speakers. In the name of the organizing committee, I express my gratefulness and appreciate your generous participation. Thank you. Thank you in addition to all the students that participate in poster section, a fundamental activity of this meeting. I want to say thank you to our sponsor, INMEGEN, in particular to Dr. Luis Alonso Herrera Montalvo, the director of our institution, in addition the director of the Department of Research at the same institution, Dr. Alfredo Hidalgo. I want to say thank you to technical staff that support us with invaluable help to handle all the media and artistic designs required to proceed with the meeting. I want to say thank you to the Red de Apoyo a la Investigación and Centro de Ciencias de la Complejidad. Both institutions belong to the UNAM. With this, let's close the meeting. Hopefully next time the CISBio MX5 be held at INMEGEN. Let's see how evolved this pandemic situation that currently we are facing. However, I am sure that we will have the opportunity to talk by electronic media and physical interact in the near future. Thank you so much and see you soon.